a quiz today. Yeah, we'll have it on Saturday instead because lots of you have sent a message that it would be better if we have it on Saturday. Okay, so let's continue. So we we continue with the brain. And before we start with the brain, we have to take a look a little bit at the anatomy, at some of the more important structures. So cerebrum, you remember, right? So two hemispheres, right and left hemispheres. Very important functions are the higher functions, the interpreting touch, vision, hearing, speech, reasoning, emotion. And I guess the most important thing is, of course, control of the movement. Control of the movement is initiated by a very specific gyrus, right? So this one, the precentral gyrus, right? This is the central sulcus. This is the precentral gyrus. This is the postcentral gyrus. The precentral is the primary motor area. So that's where the control of movement will be. The postcentral gyrus, this is the primary sensory area will be. That's where all of the ascending tracts are going, right? They're going to this area specifically. Cerebellum, we have talked about it on the previous lesson. You remember that its most important function is proprioception. This one word talks about all of these functions, right? So coordination of movement, posture, balance, etc. Brainstem, we will divide it into two. We'll talk a little bit about it today, just a bit, and most of it we'll cover next class. Brainstem is the autonomic functions, the automatic autonomic functions, so such as breathing, heart rate, body temperature, and etc., etc., etc. Could you remind me, how do we call the vomiting center? That is going to be located. Very good. We call it area postrema. Which kind of organ is it? Not organ, which kind of area is it? This area belongs to which types of organs?
Circumventricular.
Circumventricular.
Very good. Circumventricular. They surround which ventricle specifically?
Third of
But this vomiting center, it surrounds which ventricle specifically?
Fourth.
Fourth, because where is vomiting center located?
Rhomboid fossa.
Where the medulla oblongata will be, right? So right there where medulla is located, we have there the rhomboid fossa, very good. And that's where the fourth ventricle will be. Okay, what is so special about circumventricular organs?
They lack something.
They lack
brain barrier.
blood-brain barrier. Okay, so BBB is not present there. So blood-brain barrier will not be there for sure. So they lack that. Okay, so we have talked about the brain, that it consists of two hemispheres. We have the left and the right hemispheres. But you should remember that these hemispheres are going to be connected by a bundle of fibers that are going to be known as the corpus callosum, right? So corpus callosum is right here. It is just connecting the two parts of the brain. Do not forget that brain is kind of responsible for the opposite sides, right? So we have, for example, here the right side, right? So it's going to be responsible for the left side of the brain. The left side of the brain is responsible for the right side of the body. So that means that all of the fibers which are coming down from the brain, how do we call this? How do we call this?
Contralateral.
But what are they doing? Do you see they're going to the opposite side? How do we call it in neuro?
Crossing.
Decussation.
Decussation. So don't forget the decussate. Sorry, guys. They decussate. Okay, so they're crossing to the opposite side, right? So typically, as you remember, for the motor tracts, all of the decussation will be happening right there, right where the cerebrum is, right? For the ascending tracts, for the sensory, decussation happens somewhere in the spinal cord, right? Because they're going upwards there. very important to remember that typically in majority of the population, the left hemisphere is the dominant in hand and language. So our language center is located in the left hemisphere for majority of the people. Okay, this slide is very important because for sure they're going to ask you in both in neuroanatomy and in neurohisto, what are the functions of the lobes? We kind of touch them, but not too much, right? So frontal lobe, you can see their functions right here. So personality, behavior, emotion, judgment, speech, specifically the Broca's area, right? You can see that Broca's area, the Broca's speech center is located on the frontal lobe. The body movement, because the precentral gyrus is part of the frontal lobe, right? So precentral gyrus is part of the frontal lobe, so of course the movement will be there. The parietal lobe, okay, that's where the sensory strip will be. That's where the post-central gyrus will be. You can see that it's just kind of interpretation, right? So its main job is just interpretation. It's just kind of trying to understand what are you looking at. Occipital lobe is actually looking, right? For sure, you remember from your previous knowledge that that's where the vision will be. And temporal lobe is very important because that's where the Wernicke's will be, right? So Wernicke's speech area will be. We'll talk about it today. So what is the major difference between Broca's and Wernicke's area? The Wernicke's area is responsible for understanding the language. So how will the person speak if this area is damaged? Will this person speak normally or abnormally?
Normally.
Very good. So the person will be speaking normally. The problem is that they won't understand your question. So you will tell them, when were you born? And the person whose Wernicke area is damaged, he will say, my chicken was born on the kitchen, I don't know, in Malaysia. It's just a bunch of words together that make zero sense in a sentence. Yeah. So the words are coming out very nicely. You can understand what he is saying. It's just that he or she does not understand your question. The opposite is if the Broca's area is damaged, because here this patient fully understands your language, right? He fully understands your question. He just cannot answer. Okay, so the words are not coming out because this frontal area is more for the motor, right? So that's the motor area right here. So the patient will not be able to say, okay? So he starts saying, but nothing comes out. But you can see that he fully understands what do you want to talk about. How do we call it when there is loss of speech or disturbance? We call it aphasia, right? So when you can't speak.
I have
So
a question.
for sure, just a
For
second.
the-
So for sure, it's going to be in the neurohisto and in neuroanatomy, especially with Short Zolli. He loves this. Okay, so he will ask you the main differences between Broca's aphasia and Wernicke's aphasia. Yeah.
Yeah, you said that if the Wernicke's area is damaged, they won't be able to understand.
Yeah.
But are they aware of what they're saying? Just curious.
That's a good question. Are they aware of what they're saying? Not really, okay? Because for them, what they're saying makes sense. Okay, so they don't understand what they are saying. Okay, so like it's the same. Like everything is very confusing for them. They don't understand your question, nor when they speak. Like, you know, for them it makes sense. For you it doesn't, but for them it's a perfect sense. It's the same as, for example, we haven't talked about it on the previous lesson, but it's a very special symptom of cerebellar dysfunction. You ask a person to draw a clock. And how do you draw a clock? Well, you draw a circle, right? And then you say that there's like 12 here, 1, 2, 3, 4, 5, 6, and you get the idea, right? So 7, 8, 9, etc. The person with cerebellar damage, because they have a disturbance in proprioception. They have a disturbance of understanding how things are. That's how they draw the clock. And when you point out to them that half of the clock is missing, they don't understand you. They look at it, and for them, it looks normal. Okay, so that's why in neuro, it's very interesting that the people who have problems with perception, right, and proprioception, everything for them is normal. So if a person has something in his Wernicke's area, he in his head speaks perfect and he doesn't understand why you're laughing. Okay, so when you reach the 5th year, they will tell you that don't laugh, like try to do something like this, okay, but try not to smile, try not to laugh, because for them it's like, why are you laughing, like, right? It's like if I'm talking to you right now and you all are laughing, like, I think I'm saying pretty smart things, so why are you laughing at me? So the same idea is here. So for them, everything is great. Okay, very important question from Short Zoli is this. You remember we have said that for majority of the people, the speech centers, the Broca and the Wernicke area, they're dominant on the left hemisphere, right? But what if I'm left-handed, right? So what if I'm left-handed? For them as well. So only 1/3 of people who are left-handed, their speech centers may be located on the right hemisphere, only 1/3 for them, because you remember that we just talked about it, that each hemisphere is for the opposite side, right? So for motor functions, each hemisphere is for the opposite side. So if majority of people have a dominant hemisphere as the left one, right? What if I'm left-handed? That means that my dominant hemisphere should be right? But it's not the case. It's only in 1/3 of people that are left-handed. Majority of them will still have the dominant hemisphere as the left one. Okay. Here we're talking about some additional areas, so like hypothalamus, pituitary, etc. Pituitary, you remember, right? So pineal gland, you remember. Let's talk about these ones. The hypothalamus, we're going to talk right now a little bit more details about it because there are so many questions coming out in neuroanatomy and neurohisto. It's the gland which is responsible for HH. Okay, HH is hormones and homeostasis. Okay, so hypothalamus is the most important nucleus, right? It is basically a nucleus, and its major job is homeostasis. So it keeps everything at balance, such as your hunger, thirst, sleep, sexual response, right? Blood pressure, emotions, body temperature, and most importantly, hormones. Thalamus, which is a bit above the hypothalamus, is more of a relay system, okay, relay station. This is just the area where all of the fibers pass through, both motor and sensory. Okay, so it's a relay station. Majority of the tracts, majority of the axons, both motor and sensory, are just passing through this area. Basal ganglia is very special. We'll talk about it today. There are a bunch of nuclei that make up the basal ganglia. Its most important job is to coordinate fine motions, such as the fingertip movement. So this area is definitely more towards motor than sensory. So basal ganglia has something to do with the motor function. Just remember that it's super fine tuning. Like cerebellum, it makes your movements very smooth, very coordinated. But tiny movements like your fingers and your toes, that's the function of basal ganglia. Okay, so they're going to be kind of fine tuning that area. Limbic system is my favorite because that's the system which is responsible for making you who you are. That's your emotions, memory, learning, etc. Okay, we'll also cover it today just a bit. Okay, now we reach the boring part. This is Short Zoli's and Professor Wolf's favorite question. What are the borders of hypothalamus? Okay, you have to describe the borders of hypothalamus. So everything which you see here, I'm going to be describing on this picture. Okay, so that's your hypothalamus right here. That's this whole thing right here. That's hypothalamus. That's the pituitary gland you can see, right? So that's the infundibulum, which connects you to the hypothalamus, right? That's where the aqueduct of Sylvius is located. How do you call this?
Corpus callosum.
This is the corpus callosum, the thing that connects the two hemispheres together, right? And here you can see the fornix. So most importantly, the anterior border of the hypothalamus is lamina terminalis. Lamina terminalis is this area. It's a protrusion of gray matter, okay? It's just protrusion of a gray matter that is located somewhere above the optic chiasm, right? So it is located above the optic chiasm because somewhere here the optic chiasm will be. So anterior border is lamina terminalis. The superior wall is of course going to be the fornix. Okay, so the superior border is going to be the fornix. That makes perfect sense, right? So fornix is just some kind of white fibers. It's a white matter structure. It has some kind of C shape, right? And because it has a connection to the hippocampal region, its function has to be related to memory. Okay, so the function of the fornix is definitely going to be related to memory. Posteriorly, there is the periaqueductal gray substance. Okay, so somewhere at the back, we're going to have here the gray substance, and this gray substance is next to the aqueduct of Sylvius, right? And the most difficult border of the hypothalamus is the inferior surface, because inferior surface is very special. There are several areas that make it up. So infundibulum and pituitary, okay, so they are making up part of it. Mammillary bodies, they are making part of it. And a little bit of the pons, okay, so pons will also be responsible for creating the inferior border of the hypothalamus. So 3 areas make up the inferior border of hypothalamus: the pituitary, mammillary bodies, and the pons. Mammillary bodies are very special. They're going to ask you a question about them. They are white matter structures. They belong to the limbic system, and they are responsible for memory as well because of their connection to hippocampal region, the same as fornix. It's just that fornix is at the top, but mammillary bodies are at the bottom, okay, and they will be related to the hippocampal region, okay. So hippocampus, as you remember, is going to be a bit like behind there. So both of the structures, fornix and mammillary bodies, have connection to hippocampus, hippocampal region. As a result, both of the structures are related to memory. Okay, there is very important thing about mammillary bodies, which is in alcoholics, they can make up memories because majority of them are in alcoholic state for a very long time. So they forget what's the reality, what's the alcoholism, you know, like the drunk high. So they can make up the past, they can make up the memory. And this happens in mammillary bodies. So in alcoholics, mammillary bodies are creating new memories. It's like you will ask them, you will ask an alcoholic person, are you still married? And he will say, yeah, even though he got divorced 30 years ago. But his mammillary bodies erased the divorce memory and created a memory where he continued living with his wife for the next 30 years. Okay, so it's a false memory. And this is why it's very interesting, right? So mammillary bodies are part of the limbic system. That's why I told you it's my favorite, because it is so not understood. Like, how exactly does this happen? That these little bodies, these white matter bodies, are creating a new memory, and not in anybody, just in alcoholics, just under the influence of alcohol. Okay, laterally, there will be optic tract. Okay, when we will do the eyes, I will show you that hypothalamus is here, right? There are two eyes here, and the two optic tracts are literally crossing behind as well. Okay, so they're going to be crossing behind the hypothalamic area. So laterally, the optic tracts are going to be present there. Now, generally speaking,
Is it only me or?
No, I can't hear her either.
Neither can I.
Neither can I. Is she still here? No, oh, she's here.
Okay, sorry guys. Yeah, something happened. Are you still there?
Yeah.
Yeah.
Okay, sorry about that. Where did you lose me?
After you finish talking about the optic tract.
Okay, so not too far along. Okay. Okay, so laterally we have the optic tract, and that's, I guess, the most important thing. Now, I want to make sure that you guys know additional name of infundibulum. Do you guys remember infundibulum? It was connecting the hypothalamus to the pituitary gland. Am I recording? Yes. Okay, so it is connecting the hypothalamus to the pituitary gland. The other name for it is tuber cinereum. Okay, so an older professor may want you to know this particular name in neuroanatomy. Okay, so that's its other name, tuber cinereum. This is why there is an area which is going to be known as the tubal area. Okay, so this area right here, that's the tubal area of the hypothalamus. Because another name for infundibulum, I repeat, is going to be the tuber cinereum area. So on this picture, you can see that there are a bunch of nuclei present here, right? All of these nuclei are hypothalamic nuclei. And hypothalamic nuclei are arranged into different parts. So there is infundibular region. It's this one. Another name is tuberal region. That's infundibular region. There is going to be mammillary region, that is somewhere here, okay, that's the mammillary region. There is going to be prechiasmatic region, it's this one, right? So it's kind of in front of the optic chiasms. So the anterior region of the hypothalamus that is going to be above the optic chiasm is known as the supraoptic area, or its prechiasmatic region. It's identical. So the two names are the same. They're describing the same thing. The nuclei which are present in this area of the hypothalamus, you already know them. The supraoptic, paraventricular. We've seen them last semester. Just two more new ones, the suprachiasmatic and anterior hypothalamic. The supraoptic nucleus, you remember, it was making the two hormones, the vasopressin and the antidiuretic hormones, right? So the ADH. These hormones were stored in the posterior lobe of the pituitary gland. The anterior hypothalamic nucleus is responsible for thermoregulation. It is responsible for cooling your body. So the axons which are originating in this nucleus are going to be synapsing with the neurons next to your sweat glands. Which sweat glands are responsible for cooling? Could you remind me?
Eccrine.
Perfect. Eccrine sweat glands, because eccrine sweat glands are present everywhere in our body and their sole purpose is cooling. So anterior hypothalamic nucleus has to supply or innervate, right, the eccrine sweat glands. This preoptic nucleus, okay, is going to be involved in the eating and reproduction. That's why both of these things bring you pleasure. Okay, eating food and reproducing is a pleasurable events. So they are regulated by the preoptic nucleus. Suprachiasmatic has something to do with the circadian rhythm. Because it is above the optic chiasm, it has connection to the optic chiasm. So, you know, light inhibits sleeping. So suprachiasmatic nucleus has to participate in that as well. Paraventricular nucleus will also be responsible for making ADH. Okay, so it also makes ADH. And it has some functions as in stress and metabolism. Okay, so if I were you, I would definitely spend some time on this slide and try to get familiar with the functions of these hypothalamic nuclei. The central part of the hypothalamus is above the infundibulum, right? So that's why we call it the tuberal area or infundibular area. It's the same. The nuclei here are very much the same. So supraoptic and paraventricular also belong to this area. They're also located centrally. But now you're going to see that there is the arcuate nucleus, right? And there's also going to be the dorsomedial and ventromedial nuclei. The ventromedial area is responsible for eating and satiety. This is a question in the exam, okay? So ventromedial hypothalamic nucleus, what is its job? Satiety. The arcuate nucleus is responsible for orexigenic peptides. This word is crazy, but we have to know its meaning. What are these? What are they making you?
Hungry.
Hungry. Is satiety and hungry the same thing? No. So ventromedial nucleus and arcuate nucleus are located in the same area of hypothalamus, but they have two different jobs. One of them is making you full, one of them is making you hungry. Orexigenic peptide, it's the appetite stimulant, okay? In simple English, it's a stimulant for appetite. So remember that these two nuclei are sitting next to each other, but they have the different functions. Posterior region, okay, we have talked about that here. We're going to have the most important thing is mammillary bodies, okay? Mammillary bodies are responsible for memory. Don't forget the alcoholics, right? That they're very special. They have very special relation with mammillary bodies. And also we have here very interesting nucleus, the posterior hypothalamic nucleus. So this posterior hypothalamic nucleus also has something to do with the orexin peptide. So it also has to control the feeding behavior. So also it is responsible for making you hungry. But you can see that it's also responsible for the gastrointestinal motility as well. Okay, so this posterior hypothalamic nucleus also is responsible for that. Okay, mammillary, I told you it has to be memory. Why memory? Because hippocampus. Okay, let's go to the next part. Okay. Did you guys register for vaccine?
Yeah.
I took it back home.
I took one today. I had to lie. I took a COVID test and it was negative, but I had to lie that I did not have fever because that's my only opportunity. So I might die tomorrow. I'm not sure why it
Is
did
this
ask
why you were
about the
coughing?
fever, but I actually took one today. Like I went and I got vaccinated, guys, today. So I'm surprised that, you know, even though it's the shittiest vaccine out of them all, so far no side effects. Okay, so no side effects. Yeah, it's the AstraZeneca. It's the shittiest of them all, the worst of the four. But yeah.
It's so
Is this
far,
why you
right?
were coughing?
No, I'm coughing. I've been coughing for the past, like, I don't know, for the past 10 days. And I had a fever and I had a runny nose and I had a huge headache. And I remember I had to cancel because my friend has COVID. So I was sure I had COVID. And then I did the test and it is negative, you know? So that's why I was like, okay, I don't have COVID. Okay, so what do I have then? But yeah.
Maybe it's just a cold.
I know, but you know, like in this area right now, we can't cough, we can't sneeze because automatically everyone assumes that you have COVID. So as I was receiving the vaccine, I was like making sure that I'm not coughing, yeah, because I have the strong urge to cough for the past 10 days. But yeah. Okay, guys, so neurohisto part 4. So we are talking about the brain itself. Before we talk about the brain, I want to get back to the peduncles because they're very important, the cerebellar peduncles. Okay, so I see so many questions in the past papers about them. I hope you remember that there are 3 of them, the superior, middle, and inferior. Which one is the biggest peduncle?
Medial.
The middle. And with what middle cerebellar peduncle works?
Pons.
It works with pons, right? That's its only basically interaction with. The inferior cerebellar peduncle, as you remember, are receiving input from the afferent sensory fibers of vestibule, spinal cord, tegmentum, and medulla, right? So don't forget the 4th one is medulla. I also pointed out that last class. Okay, and the superior cerebellar peduncle is the output to the cerebral cortex via thalamus, right? So I hope you remember this from the last class. So if we take a look at these 3 peduncles, which one is the only one that can send fibers to the cerebrum, to the brain directly? It's going to be the superior one, but it doesn't send them directly. It sends them via the thalamus first, right? So remember we said function of thalamus, it's a relay station. Majority of fibers pass through it before they go to the brain. Okay, this is a repetition. I feel like we have talked about it so many times, so it's even pointless. You remember the difference between the nuclei, ganglion, and etc. So cerebrum is going to be a bit opposite from the brain, right? Because here the gray and the white matter are switched. As you remember, in the spinal cord, the gray matter was the inner and the white matter was the outer. How did we call the white matter in the spinal cord?
Funiculi?
It was called funiculi. We had 3 of them, as you remember, anterior, lateral, and posterior funiculi. What was in the funiculi? Say better.
Fascicles.
Fasciculi, very good. In the funiculi, we had fasciculi. So funiculus was the entire area, but the individual tracts which are present in those funiculi would call them fasciculi, right? So here it's going to be a bit the opposite. So the gray matter is the outer and the white matter is the inner, right? Generally speaking, the cortex, so the gray matter of the brain, could consist of 3. Okay, there are 3 types that could be found in the brain. There is the neocortex. Neocortex possesses 6-layered structure. More than 90% of the entire outer covering of the brain is neocortex because it has these 6 layers. Paleo and archaea cortex are very special because they do not possess the 6-layer structure. Instead, they possess the 3 layers. As a result, together, paleo and archaea cortex are known as the allocortex. Okay, so together they're making up a cortex which is called allo. Archicortex is only present in the hippocampus. Paleocortex is present in the olfactory area and someplace else. I forgot where. Okay, so olfactory area and somewhere else. Okay, I will refresh my memory, guys. Sorry, I don't remember right now. There was a question in the past paper. I will look at it and I'll put it next class. Okay, so olfactory area and somewhere else. That was the correct option. So paleo is going to be there. So don't forget the major difference between the neo and the allo is the number of layers. Because neocortex is the dominant one, more than 90% of the cortex consists of 6 layers. It's also known as the homogenic because pretty much majority of the cortex consists of these 6 layers, right? The paleo and the archi are known as the hetero because they're special, right? So they do not possess the 6 layers, they possess the 3 layers. On this picture, you can see the most important neurons in the gray matter in the cortex. Okay, so the most important neurons in the gray matter in the cortex. So here we have a pyramidal cell. We have a bunch of stellate, fusiform, Martinotti, and horizontal cell. So this picture shows you a very good representation and arrangement of these neurons. You can see that pyramidal cell is more or less at the deeper level, right? And the rest of the cells are at the top. So pyramidal cells are going to be the most dominant neurons in the brain. They are called pyramidal cells because they're shaped as pyramid, of course. Their size, is approximately from 10 micrometers to 70 micrometers. Okay, so that's the average size for them. The biggest pyramidal cells are called bed cells. Their size is more than 100 micrometers. Okay, so they're not your average size pyramidal cells, they're a bit bigger. And these pyramidal cells are very special because you remember we talked about them quite sometimes. What was their function?
They are upper motor neurons.
They are upper motor neurons. These upper motor neurons, what do they do? What is the job of the upper motor neuron?
They send impulses to the spinal cord.
So they send an impulse to the spinal cord. In the spinal cord, we have the lower motor neuron. What is another name for lower motor neuron?
Alpha motor neuron.
Alpha motor neuron. And alpha motor neuron innervates
Skeletal muscle.
Skeletal muscles.
So you remember that basically your skeletal muscles, they wouldn't move. I wouldn't do this movement right now unless bed cells tell me to do so. Okay, bed cell initiates this whole thing. What will be the location of the bed cells? Will they be present everywhere in the cortex of the brain?
In the motor guidance.
In the motor gyrus. What is another name for motor gyrus? Precentral gyrus. Bed cells are special pyramidal cells. They're motor neurons. They're upper motor neurons. The motor neurons could not be everywhere. They cannot. They're only located in the precentral gyrus, in the motor cortex, okay, in the motor cortex. So bed cells, which kind of neuron are they?
Excitatory?
By which kind of neuron? I mean like the function. There are 3 types of neurons from function perspective.
Multipolar.
They are multipolar, but your multipolar is the shape. I'm asking about the function. There are 3 neurons from functional perspective: sensory, motor, and interneuron. So bed cell, which one is it?
Motor.
It is a motor neuron. What about any other pyramidal cell, the ones that are here?
Are they interneurons?
They're interneurons. But both of them have the same shape. What is the shape?
Multipolar.
Multipolar. Okay, now you can say the multipolar. Do you get it or you don't get it? The bed cells, they are pyramidal cells, but they're special. Okay, their function is very special. They're upper motor neurons. These upper motor neurons cannot be located everywhere because upper motor neurons have a very particular job. They innervate lower motor neurons. That's what they do. Okay, so upper motor neuron, that's my bed cell. Okay, it innervates the lower motor neuron. Lower motor neuron is responsible for innervating the skeletal muscle. So bed cell can't be everywhere. It has to be in the motor gyrus, in the precentral gyrus motor cortex. Because it has a function of motor, it can be interneuron. It has to be a motor neuron. But in the gray covering of the brain, there are lots of pyramidal cells as well. They aren't big. They're just 10 to 70 micrometers. Do they have any function to motor? Not really. They're just sitting there in the gray matter. So what is their function then? They're interneurons. But don't forget that the interneurons and the motor neurons have the same shape. So they look identical. They look like multipolar neurons. But how do you know that you're looking at the bed cell and not at the normal pyramidal cell? The bed cell is huge. It's more than 100 micrometers. If you find a huge cell, that's a bed cell. If you find a little tiny pyramidal cell, it's just a pyramidal cell.
I
Does
have
it
a
make
question.
sense?
Yeah. So all the pyramidal cells are interneurons except the bed cells?
Yes. Yes, all of them are interneurons except for the bed cells, because bed cells are also pyramidal cells, but they aren't interneurons, they're motor neurons, yes. Okay, if this is a bed cell, and we have said that it innervates lower motor neuron, which is present in the spinal cord. How do you call this? How do you call this connection between bed cell and lower motor neuron? So bed cell is in the brain.
Sending tract?
Corticospinal.
It's a corticospinal tract, very good. It's a corticospinal tract. The axons of the bed cells and some interneurons that are connecting the bed cell to the lower motor neuron, they're making up corticospinal tract. Could you remind me, which kind of corticospinal tracts do you know?
Lateral?
Lateral and
Anterior?
anterior. What is the job of the anterior corticospinal tract?
Muscles of the trunk.
Very good. Innervation of the skeletal muscle of the trunk. Lateral corticospinal tract innervates skeletal muscles of the
Limbs.
limbs. Do you see the whole picture or no? Okay, do you see how bed cells are related to the alpha motor neurons in the spinal cord? Okay, a question. If bed's cell is damaged, will the patient have a reflex? Will the reflex be intact?
Yes.
Yes.
Yes. The reflex will be intact. Why?
Because the reflex doesn't involve the brain.
Exactly. The reflex does not involve the brain. It only involves the spinal cord, the lower motor neuron. So you remember how the reflex works, right? So we have the sensory neuron, we have the interneuron, and then we have the lower motor neuron. That's the reflexal arch. So in order to not have a reflex in a patient, the lower motor neuron should be damaged, right? So the lower one. But we asked about the bed cell. If the bed cell is damaged, will the reflexes be intact? Yeah. And not even intact, they will be hyper reflex because they're too stimulated, because bed cell is a corticospinal tract creator. So corticospinal tract does not only excite the lower motor neurons, it's also capable of inhibiting them, suppressing them from too much of reaction. Okay, this is a pyramidal cell. Okay, so we have said that pyramidal cells are very special because, you know, they're multipolar. Whenever I show you a multipolar neuron, how does it look like? Well, it has a body, soma, it has a bunch of dendrites, and it has like one axon, right? Does it look like multipolar here? No. Okay, so pyramidal cells and bed cells also, they're very special types of multipolar neurons. Because what is this? What is this?
The cell body?
The cell body, of course. What do you think this huge thing is going up?
Axon.
For sure, that's what lots of people think. That's an axon. But you remember we talked about it. How do we know if it is an axon or a dendrite? What should we do?
Thickness.
Consistency.
Thickness, consistency, branching. So do you see? Let's compare thickness. It starts thick, and what happens to it?
Just in.
It gets thinner. Okay, it gets thinner and thinner and thinner. So this is actually not an axon. This is a dendrite, and this is a very special type of dendrite. It has a name. This is an apical dendrite. This is the top of the pyramidal cell. This is an apical dendrite. Only pyramidal cells and bed cells possess this apical dendrite. In addition, you see that it also has two dendrites going this direction. Do you see? They're going in that way, right? So for example, this one and this one. These dendrites are called basal dendrites, and typically there are two of them. There's only one apical dendrite, the one goes upward, and there are two basal dendrites. And this little tiny thing that you see here, that's the axon. Okay, axon is very, very tiny here, you can see. So that's what we talked about. We have the apical dendrites, we have the basal dendrites. Apical dendrites are very special because they possess dendritic spines.
Can we see the axons in the previous picture?
Not too much, but yeah, okay, so this little tiny thing. Okay, so this one. On here, we cannot see, this is also an axon coming down, but on ours, I think we'll see it a bit better. Guys, could you remind me the dendritic spines? Where did we see them? This
On
word.
Purkinje cell dendrites?
Very good. Purkinje cell dendrite. Purkinje cell, is it excitatory or is it inhibitory?
Inhibitory.
It's inhibitory. The pyramidal cells are excitatory. Okay, so all of them. The bed cell and the interneurons that are normal, they're also excitatory. So you have to remember that these pyramidal cells are exceptions. Typically, Professor Wolf says the more dendrites, the more dendritic spines you have, the more inhibitory the neuron will be. And pyramidal cells are also possessing these spines, these dendritic spines, but they aren't inhibitory, they're excitatory. So these cells are exceptions, okay? They are exceptions. The Purkinje cell has lots and lots of dendritic spines, hence it is super inhibitory. The pyramidal cell also possesses the same dendritic spines, but because it is an exception to the rule, it is actually excitatory, okay? Okay, long axons, blah, blah, blah. Pyramidal cells are the principal output neurons. They're the output of the brain, okay? They're the output of the cerebrum. Stellate cells. Stellate cell, also known as the granular, also known as the non-pyramidal cell. Okay, as you can see, it possesses lots and lots of different names. In your self-control test, I think you will see this one, non-pyramidal cell. Okay, when I say self-control test, I mean exam. I'm used to saying self-control test because back in the day we had self-control tests, but I mean exam, yeah? So in your exam, you will see this one, okay, for sure, non-pyramidal cells, because we have pyramidal cells and we have non-pyramidal cells. They're the principal interneurons of the cortex. They come in all the different shapes and they're typically small, less than 10 micrometers. And because they're interneurons, of course, they have to be multipolar. So this is how it looks like, right? So probably all of these things are dendrites because you can see that they're branching out, right? So these are also dendrites. And most probably, maybe this thing is the axon, but I highly don't think so because there's a bit of a branching here going on. So probably also a dendrite. Probably we can't see the axon coming down this way. They're just interneurons. Cells of Martinotti. Cells of Martinotti are polygonal. They have very short dendrites. Their axons extend towards the surface and bifurcate and run in the most superficial layer. So this cell is also an interneuron, and it is sort of upside down to the pyramidal cell. Okay, so if pyramidal cell is this way, Martinotti's axon is going to go this way. Where did we see very similar position? Where one cell was going this way and another cell was going that way.
The Purkinje cell and the granular cell.
Very good. Yes, yes, yes. Purkinje cell and the granular cell in the cerebellum, right? The same thing is here. We had the pyramidal cell kind of going down, and we have the cells of Martinotti going upwards. And you can see it also has some kind of synapses. Okay, horizontal cells of Kayal or Retzius Kayal cells. They're very prominent during development, but disappear after birth. So in our slides, for sure, we'll not have them because we have an adult brain. This is the least common cell type, always parallel to the surface, always in the most superficial layer. Okay, but it's a more of an embryological cell rather than an adult cell. Also interneuron, so just synapses with pyramidal cells. The staining that we have, the staining is known as a Nissl staining, which mainly stains DNA and RNA. So is it a basic or acidic dye?
Basic.
It is a basic dye. Why?
It'll be positively charged. It's going to be staining a negatively charged substance.
Very good. So DNA is highly negative. RNA is also highly negative because of the phosphate groups, right? So Nissl, if it stains the DNA and RNA, it has to be a basic dye. So it has to be positively charged to stain these things. Two gyra are visible. We talked about them, right? So central sulcus, the precentral and the postcentral gyra, the motor and the sensory. According to the exam, you generally have two types of cells in the brain, pyramidal and non-pyramidal. But don't forget, non-pyramidal are all of this. The Kayal, the Martinotti, the stellate, they're all considered to be non-pyramidal cells. So what are the 6 layers, right? Because this is the neocortex. What are the 6 layers? The top layer is the molecular layer. This is where you will find Kayal cells in embryo. Okay, so this is where the cells will be. We don't have them much, okay? So they aren't dominant in the adult. External granular layer, this is where the non-pyramidal cells are going to be dominant with the stellate, okay? So granular non-pyramidal cells, that's where they're going to be here. External pyramidal cells, that's where we have the tiny pyramidal cells sitting here, right? So tiny interneurons are sitting in the 3rd layer. Internal granular layer, we have here a bunch of interneurons, right? Like non-pyramidal cells, but we also have a very special band here. This band is known as the outer band of Bay-Larger. So in this 4th layer, you're gonna see that that's where the outer band of Bay-Larger will be. I will discuss it right now. Okay, the next layer is the internal pyramidal layer, right? The 5th one. That's where the Martinotti cells can be sitting with some additional pyramidal cells. And the last layer is the multiform. Multiform, in theory, could possess all the cells. Okay, you could find their pyramidal, you could find their Martinotti, you could find their stellate, granular, retius if you're an embryo, and etc. So in the multiform, it has a name like that because in theory, all of them could be there. In the internal granular layer, internal pyramidal, it's not granular, in internal pyramidal layer, okay, you're going to see that there's additional band here, and it's called inner band of Bay-Larger. So there are two bands of Bay-Larger visible here, yeah? The outer is in the internal granular, and the inner is in the internal pyramidal layer. So these bands of Bay-Larger are axons. Axons are white matter, but these 6 layers are making up gray matter. So in the brain, you can see that in the gray matter, in the outer cortex, there are axons running, okay? And these axons belong to certain cells. We'll discuss them right now, right? So these axons are making up the bands, the band of Bay-Largers. Now, if you can see here, I'm highlighting to you large pyramidal neurons. Which cells could this be?
Bed cells.
Bed cells. So if you were to have bed cells, in which layer would you put them? Take a look where we are.
Internal pyramidal.
Yes, in the 5th layer, okay, in the internal pyramidal. So if bed cells are present, the only layer where they will be is this layer right here, in the internal pyramidal layer. If bed cells are present in this particular layer, then that layer changes its name. Then it is called ganglionic layer. So if bed cells are sitting here, then you will call it ganglionic layer. Can bed cells be everywhere? Like, let me rephrase it. What is the only place where you could have bed cells?
Precentral gyrus.
The precentral gyrus, the motor gyrus, its 5th layer will possess bed cells. Everywhere else, it won't. So ganglionic layer is only present in that area. It's only present in the precentral gyrus. Does it make sense? So because ganglionic layer possesses bed cells, and bed cells are only present in the motor gyrus. Yeah? Okay, so how to differentiate between the pre and the post-central gyrus? You look for the bed cells. You look at the 5th layer. Okay, you look at the 5th layer. If you see huge pyramidal cells, then that's it. That's the bed cells, because in the sensory, they will be absent. It's a very good picture of the bed cells, right? You can see that. How do we call this?
Apical dendrite.
Apical dendrite. This one and
Basal
this
dendrite.
one are basal dendrites. You can see that there's only one apical dendrite and typically 2 basal dendrites. And this structure right here is going to be an axon. Okay, this is the axon right here. Okay, so we have talked about it. So this is the additional information, but we talked about it, yeah? Let's talk about the Bay-Larger. Okay, so this particular band. So basically, we have said that we have 2 bands of Bay-Larger, right? We have the outer and we have the inner. The pyramidal cells of the external pyramidal layer send their axon down, and as they send it down in the internal granular layer, this axon turns 90 degrees and starts to run tangentially. So this turn of axon of these cells creates this band. So band of Bay-Larger, we have said it consists of axons, but whose axons specifically? The pyramidal cells, okay? So the pyramidal cells of this area. The outer one specifically, I'm talking about the outer one, okay? So the outer Bay-Larger band. It consists of axons of the pyramidal cells, and these axons are running tangentially. And I hope you remember how tangential looks like, right? So do you see? It was like an axon was going like in a cross, in a radial kind of manner, right? And then it does this, okay? So it turns, there is a 90 degrees turn of the axon. So this tangential direction creates the external band of Bay-Larger, so the outer one. But the inner band of Bay-Larger is not created by the pyramidal cells. It's created by the granule cells, okay, by the non-pyramidal cells. They send their axon as well, like this. Does it make sense? So this is the outer band of Bay-Larger. This is the inner band of Bay-Larger. The outer band of Bay-Larger is located in the internal granular layer. But the axons that make it originate in the external pyramidal layer, the one above. That means that a cell that has to send the axon down one layer has to be pyramidal cell, right? It sends its axon down, 90 degrees turn, creates the outer band of Bay-Larger. The inner band of Bay-Larger is located in the internal pyramidal layer. But where is the cell sitting that is sending their axons down this layer? It's sitting in the granular layer. That means that the granule cell, the stellate or non-pyramidal cell, sends its axon down, also 90 degrees turn, tangential. So these two bands are formed by axons that are running in the same direction. But who gave you the axons? Who gave you the axons? For two bands, it's different. The axons in the outer band were given by the pyramidal cell because that cell is above. The axons in the lower band were given by the non-pyramidal cell because it is above. Yes, no? Okay. We talked about ganglionic layer, right? That is a very special layer because it has bed cells. So this layer is not present everywhere. It's only present in the motor gyrus, in the precentral gyrus, right? Multiform, we talked. Okay. So this is the same idea, right? So we have a bunch of layers here. So make sure you remember, right? So the top one is the molecular. As you can see, it's very pale because we don't have here much cells. External granular, external pyramidal, internal granular, internal pyramidal, multiform. I repeat, this one is not present everywhere, just in the motor. Okay, bed cells, we covered them. Do you understand that they're the upper motor neurons, right? They innervate the lower motor neurons, right? Size, we talked about them. The axons that they make up are corticospinal tract. Okay, what if a bed cell doesn't go to innervate lower motor neuron? We said that bed cell should innervate lower motor neuron. The alpha motor neuron is the same name for it, right? So alpha motor neuron. And then this lower motor neuron will innervate the skeletal muscle. We said that the axons of the bed cells coming down to innervate this neuron create corticospinal tract. What if bed cell doesn't go here? What is the possible location it could go? To rephrase the question, where else can bed cell go What else can it innervate? Lower motor neuron via the corticospinal tract for sure, but there's one more thing it can innervate. What is that?
Corticobulbar.
Amazing. Corticobulbar. What does corticobulbar innervate?
Brainstem.
Nope. Via the brainstem for sure, yeah. But what is the final product?
Vision and
The face. Okay, so all of the facial nerve is going to arise from the bulbar region, okay, pons, that's the area where a facial nerve will be. So the bed cell will send its axon to the brainstem area for sure, and from that brainstem area you're going to innervate the face. So this is the corticobulbar, okay, so from the brain to the brainstem, this is the corticobulbar. From the brainstem to the face, that's your cranial nerves. So trigeminal, facial, and additional ones, okay? So for the face, okay? So that's the ones that are going to do so. So bed cell does not only have to innervate the lower motor neuron, it has a ability to innervate the brainstem as well. Okay? Okay. Okay, in the brain, we have very special, interesting tracts. Okay, so we talked about tracts in the spinal cord. Let's talk about them in the brain. There are three types of them. We call them projection fibers, association fibers, and commissural fibers. Projection fibers interconnect cortical and subcortical structures. In other words, they connect the outer gray matter with the inner white matter. So, which ones are belonging to the projection fibers? This is going to be the internal, external, and extreme capsules, the same as the optic radiations. Okay, so these four, they are projection fibers. I guess if I were you, I would focus on internal capsule because internal capsule is very special. Through the internal capsule, corticospinal and corticobulbar tracts are passing through. So inside of the internal capsule, you can see axons going down, okay? And these axons belong to the corticospinal and corticobulbar tracts. So internal capsule is a projection fiber because it connects the two areas, right? The association fibers are going to be connecting different parts of the cortex within the same hemisphere. So, for example, from the frontal lobe of the cortex all the way to the parietal, okay? So it stays within the same hemisphere. Very interestingly, the bands of Bay-Larger are going to be belonging to the association fibers because these axons are traveling within the same layer, within the same hemisphere. Surprisingly, you guys don't talk about it in the second semester. Okay, I was going like second year, sorry. Okay, I was going through the lectures, barely. They gave you this particular area and that's it, okay, that these tracts belong to association fibers. And then I sent an email to Shortzoli. I was like, is there anything particular you guys need to know about them? He said, no, okay, you guys don't have to know about them. But in fifth year, they're very important. So when you reach the fifth year, you'll have to relearn a couple of things. So for the sake of the exam, the only thing worth mentioning about association fibers is that these axons are within the same hemisphere. Okay, they're within the same hemisphere. And maybe you should remember that the bands of Bay-Larger are considered to be them. Commissural fibers are very important. That's the one that they want you to know. Because commissural fibers connect to different hemispheres. So if association connects the same, connects different areas within the same hemisphere, the commissural ones connect different hemispheres. The ones that belong to it is, of course, corpus callosum, anterior commissure, and posterior commissure. Corpus callosum is very interesting structure. There's so many different researches about it. What does it do exactly? But for sure, it has something to do with the motor, because that's where the fibers will be crossing. Okay, so they'll be going to the opposite side. So there has been a very interesting solution to seizures, epilepsy. Do you guys know what is epilepsy, right? Epilepsy is when the brain neurons are firing too much, so you're starting to seize, right? So back in the day, to cure epilepsy, they used to cut corpus callosum. That was the solution. They would just cut the connection between the two hemispheres, and supposedly the epilepsy will heal. The problem was that in approximately 45% of the cases, the person will lose ability to move half of his body. Okay, so he won't move either left or left arm or left leg, okay, together, or he wouldn't move the right leg and right arm together. But in some individuals, there is agenesis of the corpus callosum. Agenesis is embryological misdevelopment. There are some children, some babies that grow up to be adults that are born without corpus callosum. Do you think they have problems with movement? Do you think they can't move their things? They're perfectly fine. Okay, so that's why they do not understand how is it possible. How come that if you have an adult and you cut the corpus callosum, in 45% chance that you may lose half of your body's skeletal muscle function. But a baby is born without corpus callosum in the first place, and he or she is totally fine with moving all the arms and all the legs. Okay, so that's why they are questioning, is corpus callosum really a motor structure? Does it really have a job in motor, right? Because not everybody needs it for motor. Definitely, it has something to do with problem solving and verbal processing speed. Okay, so they realize that majority of patients, more than 90%, if you cut their corpus callosum, they become like a little bit, I don't want to say a vegetable, but they are a bit slow. Okay, so they're just a bit slow. Okay, so it takes time for them to process things, to try to solve a problem, to initiate speech, to understand what you're saying. It takes time for them if you cut it. But again, babies who are born with complete absence of corpus callosum are fine in everything. They're totally fine. Okay, there's zero problem. Anterior commissure is also known as the precommissure. It is placed in front of the fornix. Okay, so it is a bit more anterior of the fornix. Two parts. It possesses two types of fibers: olfactory fibers and non-olfactory fibers. The most important thing that olfactory fibers are present there. Olfactory fibers are in the anterior commissure, but it makes sense, yeah? So fornix, that's where the sella turcica will be as well, a little bit below, right? So for sure it has something to do with the olfactory area. Posterior commissure, very important, is related to bilateral pupillary light reflex. If we have time next class, we will try to look at it, because it's not only important for neuroanatomy, it's also important for neurophysio. This thing, oh, they will drill you on this in neurophysio. Okay, so they would want you to know this reflex for sure. So how exactly do you see, right? So the posterior commissure, it possesses fibers related to vision, okay, specifically to the light reflex. Do you guys know the light reflex, right? So what happens if I shine a light to your eye?
I think it has something to do with the facial expression muscles.
What happens if I shine the light directly in your eye? What happens to your pupil?
Dilates. The
Are you
pupil
sure?
dilates. No, sorry, it constricts.
It constricts. It constricts because there's too much light. What happens if I put my hand right here, right? And I shine the light here, yes, to my right eye. What will happen to my left eye?
Also constricts.
Also constrict. That's the very interesting bilateral reflex. So in movies, they do it very weirdly, yeah? They're like tick, 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 tick. That's not how it's done, okay? So you don't shift it back and forth. You have to put your hand here, and as you shine the light in one eye, you notice what's happening to another one. Another one should be constricting. It doesn't get the light, but it constricts, yeah? Because they're connected. The two tracts at the back are connected. So they are together. The fibers that are connected are running in the posterior commissure. I really like how this particular phenomenon is called. It's called consensual light reflex. Okay, so when you do this consensual, it's like one agrees with another. Yeah, it's like, are you constricting? Okay, I'm also constricting. Okay, are you dilating? I'm also dilating. Okay, so it's consensual. Both of them are on the same page with one another. Okay, this part we talked about is just additional information. Allocortex, as you remember, are paleo and archae. They have 3 layers instead of 6. Those 3 layers are called molecular, pyramidal, and polymorphic. Polymorphic, another name is multiform. No changes. Okay, molecular, pyramidal, multiform. Okay, we talked about this. This is a very important thing for the exam. They're going to ask you what is the afferent input of the cerebrum. As you can see, thalamus, right? Majority of the fibers that are going into the sensory area of the brain, they're related to, like, they're passing through thalamus. What is the efferent output of the brain? Corticothalamic, corticocortical, corticostriate, that are to the brain stem and spinal cord. Corticostriate, that's 2 of them. That's the corticobulbar and corticospinal. It's just another name for them. Why striate? It's a very interesting question. If I were examiner, I would definitely ask this. Why striate?
Does it go through the striatum?
Aha, but why do we call it striatum?
Because of the
striated?
2 bands.
But what are the bands? What is creating this striated appearance?
Red cells.
Well, you're kind of correct. Internal capsule. The striated area, it's where the axons of the internal capsule interact the structure of the nucleus that is present there. I'll show it to you what I mean. Okay, so here, okay, so striatum. Striatum has 2 nuclei, right? So the caudate and the putamen. So these are the 2 nuclei. But between them, this area right here, that's the internal capsule. Okay, internal capsule runs between them. So these 2 are gray matter. They're nuclei, they're gray matter. Internal capsule is white matter, so it creates this striated appearance. So striatum is internal capsule, and you remember which tracts are passing through the internal capsule? Well, the corticobulbar and the corticospinal. So corticostriate fibers, it's like a uniform name for the 2 tracts, the corticobulbar and corticospinal. Do you understand? Right? So why striate? Because it's striated, because internal capsule interrupts the nuclei. Which nuclei? The caudate and the putamen nuclei. Okay, so this picture is very important. If I were you, I would spend some time on it, especially for neuroanatomy. So what we're looking at the basal ganglia. which nuclei are here. So caudate, putamen, together they're called striatum because that's where the internal capsule will be running and interrupting them. We have here the two globus pallidi. Okay, so externa and interna. Together with putamen, they're creating lentiform, right? So together they're lentiform. There is subthalamus, there is red nucleus, substantia nigra, amygdala, and this big thing here is thalamus. Okay, so you can see that there are two of them. Whenever I say there are two, people usually are like, what? But yeah, there are actually two thalamuses on two sides. Okay. I feel like I want to say something here. Okay, never mind. So caudate nucleus. So together with putamen, right, the caudate nucleus is going to create this striatum. Do not forget it's called striatum because it is separated by the internal capsule. Inside of the internal capsule, you'll find corticobulbar and corticospinal tracts. Both of these tracts consist of the bed cells axons, right? So they're coming down there, the motor and etc. Primary function of the putamen is going to be to regulate movements at various stages, preparation and execution. So whenever you prepare the movement, that's the putamen's job. Executed, also that, okay? And various types of learning. Putamen needs GABA, acetylcholine, and enkephalin. Enkephalin is not as important. GABA and acetylcholine is important. So putamen has two functions. It can excite and it can inhibit, right? It can stimulate the particular movement, like it can give a green light for the movement to occur, but it can also inhibit it because it has GABA, right? So it says no movement should occur. Putamen also plays a role in Parkinson's disease. Okay, so it does. Caudate nucleus has something to do with the spatial information, right? So, and motor behavior as well. It's very interesting that together, putamen and caudate can be impaired in the Parkinson's disease. And you know, Parkinson's disease are the jerky movements, right? So the person has a tremor. The hands are constantly possessing resting tremor. The hands, the head is almost moving, always moving. But not everybody knows that another very interesting syndrome of Parkinson is that they have a loss of spatial memory. What do I mean? If I ask you right now, how do I get to Yellow Church from your flat? You immediately start to visualize, yeah, like for me personally, I'm going to go outside, turn left, turn right, tram number 1, 5 stops, Yellow Church. Yeah, so I have a spatial memory for that. I remember how to get there. People with Parkinson's disease, if their caudate nucleus is impaired, they can't. They understand that they're in the hospital, but you ask them, okay, how do you get from your house to the hospital? And they're lost. They can't remember. This is why lots of Parkinson's disease patients get lost. They literally get lost, okay, because they do not know how to get home. Their spatial memory isn't working. They know how their home looks like. They know where they are. It's not like they're dumb, yeah, like they understand. Just this pathway of going from the hospital to the home is impaired. They can't because caudate nucleus is impaired. Why is it impaired? Well, because you remember that Parkinson's disease has something to do with the dopamine. Okay, so it has dopamine. It needs in Parkinson's disease there is a dopamine depletion, and you're going to see that caudate and putamen and actually majority of the basal ganglia, in order to work, they need dopamine. In order to do their jobs properly, they need dopamine. Who supplies the dopamine? Who out of this is going to be creating dopamine for the use by the rest of the basal ganglia? The substantia nigra. Okay, so that's the area of the basal ganglia that has one job, and one job only, is to create dopamine, to secrete dopamine that can be used by the rest of the nuclei. Caudate nucleus is also very important for your posture, okay? And we talked about that it works with the substantia nigra because it needs the dopamine. Globus pallidus, we have two of them, externa, interna. Never ever have I ever seen a question about them. Okay, so they communicate with the subthalamic nucleus and they can inhibit the thalamus. That's it. Okay, there's nothing more that you need to know about them. So just communicate with the subthalamic and can inhibit the thalamus. Substantia nigra is important. You need to remember that it actually has two parts. It has the compacta and reticulata. If I have to show it here, right, so this is the substantia nigra, you're going to divide it in two, like this. And the one closer to the red nucleus, this is going to be the compacta. The one under it is the reticulata. Okay, so substantia compacta is at the top, reticulata is at the bottom. We said that substantia nigra secretes dopamine. So which part exactly secretes dopamine? The compacta. So in Parkinson, which part degenerates? The compacta. Okay, so the pars compacta of the substantia nigra, it definitely degenerates in the Parkinson disease. Why? Nobody knows, right? They don't know. Red nucleus, you remember. Rubrospinal tract. What is its job?
Crawling.
Yes, crawling. So in babies, that's a very important nucleus. It's the important nucleus for the motor in babies, right? But you're going to see that it also has some kind of function in your self as well, in the adults, okay? So maybe something to do with the fingers, okay? But I don't know, legs, it does have a job, okay? Because it is responsible for this like almost involuntary, subconscious position of your limbs. Like you don't think about them. Yeah, you don't think when to flex them, when to extend them, and red nucleus has a job for that. But if we have to say which function is the most important, for sure crawling in babies. That's when it is super active, on fire, and doing that. Why is it called red nucleus? Because it is filled with iron in two different forms, hemoglobin and ferritin. Okay, so that's why it's called red, because it possesses lots and lots of that. Why is it called substantia nigra? Because here you have neuromelanin. Neuromelanin. It's not the same as melanin in your skin and in your retina. Yeah, it's a bit different. So it's called neuromelanin. This neuromelanin is secreted by the neurons of compacta and reticulata. Okay, so this iron is also going to be present in the nuclei, in the neurons, sorry. Okay, so it's going to be present in the neurons of the red nucleus. Amygdala is my favorite. It's part of the limbic system, so it has something to do with the memory, decision making, emotional response such as fear, anxiety, aggression. This is very interesting research. You don't have to know this, but I put it there for you. Basically, in one study, they have, through electric stimulation, they realized that right amygdala induces negative emotions such as fear and sadness. And left amygdala, the opposite, induces pleasant happiness or sometimes unpleasant fear, anxiety, and sadness. So what they said, right? So right is the bad one, left is the good one. So they said that the depression, right? Depression is when it's like that. When you take drugs, it's like that. Okay, so these two amygdala, they have to be at balance. Okay, so if in different psychological psychiatric disorders, they kind of don't function properly. Yeah? So one of them is more active, one of them is less active. And one more story here that I really love. As you see, amygdala has something to do with fear, yeah, anxiety and aggression. Actually, two more stories, sorry. One story is that there is a disorder where people are born without it. They don't have it. They don't have amygdala. So what do you think? Which kind of people are these? Knowing that it has something to do with the fear, anxiety, aggression. Well, these people have no fear, yeah? They have no anxiety whatsoever. So these people, for example, I don't know, they go and they gamble their money through and they're fine, yeah? So they're not afraid of it because they don't understand the concept that this is bad, yeah? Like they go to their exams and they don't feel the anxiety that, oh my God, I might fail. They don't feel it, okay? So these people are almost like emotionless, okay? So they almost have no emotions. The second story is related to Nazi World War II. As you know, Hitler was very interesting person in the way that he was very fascinated by creating this particular army, yeah, that could like be controlled with the tip of a finger. So there is a study where he was one of his doctors, I forgot the name, they were surgically removing amygdala on poor people in the concentration camps, because you know in concentration camps there were three types of people: gays, communism, and Jews, of course. So those people have suffered a lot, and one of the research was to remove the amygdala. Basically it was almost successful because he did create fearless soldiers. Like he fully brainwashed those people through pain, removed amygdala, so no fear. The problem was that those people were a bit uncontrollable, meaning that they did not think with the emotions, yeah, they thought with the logic. And there was one particular case where basically there were 20 of them surrounded by some kind of army, I don't know, American or USSR army. So what do you do? So what would you do if there are 20 of you and you're surrounded? What do you do? A normal person who has emotions will try to protect the 19, yeah? Like you have this particular instinct to protect the person next to you. So what they did, they blew themselves up. Okay, they just committed this particular suicide. They blew themselves up, not trying to blow the other people, yeah, like the other army. They blew themselves up because that was the logical thing to do. So no fear, but it backfired, yeah, so they killed themselves basically. So that's one of the interesting things about the amygdala. So hopefully you remember about it. But so many researchers, guys, like I could talk about amygdala forever. Like you should also research about it because so many interesting, very unethical, but at the same time very interesting like studies. Okay, guys, let's do the slides. So cerebrum, Nissl staining, right? We have the two gyra. They have the precentral, the postcentral gyrus right here. Precentral is the motor, postcentral is the sensory. This area that you can see here, this is the central sulcus. This is the gray matter. This is the white matter, right? So this area right here, that's the cortex, right? And this is the white matter, the medulla. I'm not sure if you can see, but even with this magnification, I can tell you that this is a precentral gyrus. What should I do to make sure that this is a precentral motor gyrus? Which layer should I look in?
Inner pyramidal.
In the pyramidal layer? Okay, it's right
ganglion
here.
There.
Yeah, and you can see those big dots. Do you see? They're huge. Which cells are these?
Bed cells.
Bed cells. So if bed cells are present, that means that this layer becomes now ganglionic, and this is a precentral or motor gyrus, right? The next one will be the postcentral sensory because the fifth layer lacks them. All right, so let's take a look. First layer.
Molecular.
Molecular.
External.
Granular.
External pyramidal.
Pyramidal. These cells here are pyramidal neurons, right? They're intraneurons. They aren't bed cells. Fourth layer.
Internal granular.
Internal granular. Internal pyramidal. You can see that there are no bed cells here. You can see bigger neurons here. And then the last one is the multiform, right? So judging from this picture, is it sensory or is it motor?
Sensory.
Sensory, because the fifth layer, the internal pyramidal layer, it lacks the huge pyramidal cells. It lacks them. Okay, this is how the pyramidal cell looks like, right? So as you can see, our picture is a bit better than before, what we saw, but still you can barely see the dendrites and the axons. It's not very visible because this is a nisostanian. The cell body is the most visible part, right? You can even see the nucleolus. But yeah, what are these? And these. And these. These are blood vessels, guys. Okay, so they aren't stained nicely, okay, but these are actually blood vessels. Okay, so we talked about them, we talked majority of those things, so don't forget not only corticospinal tract, but also corticobulbar. The bed cells, they can also participate in the corticobulbar tract, right? So apical dendrite, basal dendrites, okay, maybe this is the axon, right? Maybe this is the initiation of the axon as well. But yeah. Golgi impregnation, right? So this is silver. The idea is just to show you again that we have here the cortex, we have here the medulla, and within the cortex, there are a bunch of pyramidal cells. Now, this is a very weird slide because on this picture, you must be able to say what I have highlighted to you is actually false. It's not a pyramidal cell. Okay, so this is a pyramidal cell. Okay, this is the cell body of the pyramidal cell. What you see here, it's more or less circular, the same as this one, yeah, and this one, and this one. And this is also circular, but this one is pyramidal. Do you see the difference, right? Circular, pyramidal, circular. Okay, circular, 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 a bit pyramidal. So the pyramidal are, of course, the pyramidal neurons. The circular ones are the non-pyramidal cells, the stellate or the granular cells. Okay, so the most visible interneurons, right? So that's them right here. A bunch of lines here are visible. That's the dendrites and axons. Generally speaking, the thicker line that you see, that's an axon. Okay, a bit thinner, that's the dendrites. So it's an axon, that's a dendrite, right? So this is more or less an axon. This is also more or less an axon right here. The rest of them here are dendrites. Crescil violet, okay, crescil violet, this magnification, very nicely visible, right? The fifth layer possesses this particular cells, very nicely seen. So these are the bed cells. So which gyrus is this?
Post-central?
Yes, okay, they're upside down. Yeah, do you see they're like mirror image a bit, right? So this is going to be the post-central, and this one is actually going to be a pre-central. Why? Because in the fifth layer, you can see this particular neurons right here, right? These are the bed cells. Clearly, they're bigger than the rest of the neurons. What is this?
Pia matter.
Pia mater, it is also vascularized, okay, in the cerebrum. I think I told you last class that it wasn't. It is. It was my fault. Okay, so it is vascularized. This is the central sulcus, so vascularized pia mater. Okay, here we have GFAP. Hopefully you remember this. What is GFAP?
Was it in the fibrous astrocytes?
Yes. Fibrous astrocyte possesses this more than protoplasmic. What does GFAP stand for? Glial fibrillary acidic protein. Okay, so glial fibrillary acidic protein. What is it? It's an intermediate filament type 3. Okay, and it is present mostly in the fibrous astrocytes and a little bit in the protoplasmic. I told you that we can use GFAP to see the fibrous astrocyte because we can make an antibody against them, right? Put that antibody to the staining, and we can see the fibrous astrocyte. So clearly, you can see that this is the cortex and this is the medulla. In other words, the gray matter and the white matter. Which matter has more color to you?
The white matter?
For sure. Okay, so the medulla, the white matter, it possesses more color. That means that this area possesses more GFAP. So who is here nicely visible with GFAP? The fibrous astrocytes. And you remember that protoplasmic astrocytes are in the gray matter. Fibrous astrocytes are in the white matter. So clearly visible right here, right? So we magnify, you can nicely see the protoplasmic astrocytes. In the gray matter, they aren't visible. Here they are, right? So you can see that these are the blood vessels, and these circles, empty circles, are blood vessels. And these brownish cells here are fibrous astrocytes. Okay, so these right here are fibrous astrocytes. So empty holes are blood vessels. Brownish cells are fibrous astrocytes. This picture, it's archicortex. Okay, so that's the hippocampal area. I send you this picture in the groups. Have you seen? It was marked. Okay, so in the groups, I put it there for you. It was marked because typically we just focus on this area, but Professor Wolf last year, he wanted more. Okay, so I marked everything for you there. And it's already in the group, so just try to put it there. So that's where the archicortex cortex will be. That's where the hippocampal area will be. That's where the 3 layers are. The 6 layers of the cortex are here. They're very nicely visible here, but the 3-layer cortex is this area, okay? The molecular, pyramidal, and multiform. What is interesting about this particular slide is this area, okay? So that's where they're gonna take you as well. That's the choroid plexus. Choroid plexus, as you remember, consists of a bunch of blood vessels and ependymal cells, right? So what you can see here, this is choroid plexus. It consists of blood vessels and ependymal cells. So these cells are ependymal cells. These, by the way, as well, because they're lining the space is the lateral ventricle, right? The space is lateral ventricle, and ependymal cells are lining that ventricle as well. Okay. Okay, so that's it for today's. Okay, so we are going to, I think, meet on